It's my pleasure to invite Zelda Mariet. Zelda is a co-founder and the vice president of research at Bioptimus. Her expertise lies in applying principled mathematical approaches to unlock insight from noisy, high-dimensional data. Zelda earned her PhD in computer science at Massachusetts Institute of Technology, then worked for several years at Google DeepMind before she became a co-founder and VP of research at Bioptimus. So let's welcome Zelda.
Thank you very much. Before I get started, I really need to know how the clicker works because I have a lot of transitions that are timed. So let me try. It is not the enter key.
I never quite figured it out.
Aha. Is it this guy?
Yes. Move forward, and if you need to point it, you may need to point it left.
Okay, perfect. Thank you. Thank you very much. Thank you very much for the kind introduction. Unfortunately, Tom already kind of answered in the very last question that he received a lot of what my first slides are going to be about. So I apologize, but I will be going through them anyways. So as Sumaya said, I'm a mathematician by training and a computer scientist by trade. So today I'm going to be taking a really computer science-based approach to thinking about AI for drug discovery and biomedicine in general. I hope you don't probe me too much on questions like HER2, but I can give it a try. However, I will be talking about transformers a lot. And so today what I want to talk about is where I see the future of the field going today at this intersection of artificial intelligence and drug discovery and biomedical computational biology research in general, and how I see the next transformations that we're going to see are going to occur in the next couple of years. And to me, and Tom was also talking about this, a lot of what we're going to see is going to come from multimodality. And so I will be talking about how you can build foundation models that start maybe at one scale, at one level of the biological domain, and then can expand to cross modalities and build more powerful tools. Excellent. So this is the part that you've already heard, but I spent a lot of time on this slide, so you have to see it anyways. Drug discovery, right, from my perspective as someone who's coming at it from computer science optimization perspectives, is an optimization problem. When I was at Google DeepMind, I actually started working on applications of machine learning to biology for drug discovery, specifically designing antibodies for COVID, actually. And so from my perspective, what I got was an optimization function that you can measure because what we're looking at is the log KD we get from a wet lab, right? And we can measure it. Wet lab technology has come a huge way. We can get high throughput measurements. So we have this function, and it's great. You measure it. You can get as many measurements as you want as long as you can afford to pay for them. And so you have what is a classical setting for machine learning optimization. The problem is that, as Tom was saying, you're not just optimizing the log KD. You have a lot of dynamic constraints that are evolving as you design your molecules, as you kind of figure out which direction you're going to go into. And in the case of COVID, also COVID itself is doing its own thing, kind of as we're trying to design treatments, which was, as you may recall, a little bit annoying. The problem is that these dynamic constraints, you can't measure them efficiently, right? Like they actually depend on the current status that you're in within your development process. Figuring out which are the next ones you're going to want to measure is hard. There, the technologies don't scale as well. They're time consuming. They take a long amount of time to go into measuring them, and they also cost a lot of money. Which means, they didn't refresh the slides, that you can't actually model them unless you have a bespoke AI model that's going to require a lot of data to train or take a lot of time from the perspective of the scientist who's going to have to embed their own knowledge of what's going on from the chemistry and biology side of things. Instead, what we did at Google DeepMind, what I was doing, and the way I thought about this was how are you going to build these surrogate targets that are going to try and get you to measuring things like expressibility, synthesizability, robustness to the Omicron variants and so on and so forth. And you can do some really cool stuff, don't get me wrong, right? Like you're gonna do things like target sister diseases. We looked at like MERS and SARS at the time, so on and so forth. and you can do stuff that I did my PhD in, so I obviously think is really cool. You can try and build in diversity constraints so that instead of optimizing by building one single molecule, you're actually trying to optimize your function to generate a diverse set of candidates, such that hopefully, even if 90% of them don't pass through these iterative sieves, at least one of them, by virtue of being different from all of the other ones that fail, will actually manage to go through the entire validation process. So there are a lot of things you can do, and they're, from a scientific perspective, really fun. The problem is that we're still in a setting today where once you get to clinical trials, 90% of these drugs will fail, right? And that is a lot of money, this is a lot of time, these are a lot of patients that need a treatment today. It's a massive blocker at the intersection of what machine learning could be doing for drug discovery and where we're at today. And I can't really, as a computer scientist, right, I'm not going to come in and say, okay, well, we're actually going to find a really efficient way of doing measurements for IC50, right? I can't do that. I'm sure somebody can, but that's not going to be me. What I can do is focus on the second bullet point, which is the modeling part, and look at this question of how can we actually build into a setting where we will have models that are going to scale and that are going to be able to actually predict all of these constraints dynamically. The trick, and I'm talking about foundation models, so the answer is going to be foundation models, is going to be having models that can take advantage, as we were just saying right before, of a huge amount of data that's typically unlabeled, and then be seeded with a very small amount of measurements. This was the Farsi example, essentially, right? If you take a small amount of measurements, they're expensive, but I mean, they exist. We have them. Drug discovery has been going on for a long time. You can seed the model, and either in a fine-tuning setting or in a few-shot setting, all of a sudden, have a model that can give you answers without requiring an avalanche of data bespoke the problem you're looking at. So what we want to do is to build this model, right? We want to build a foundation model that is going to be able to understand these different connections. Here's the thing with biology, as I have learned coming from computer science, is that it's complicated, but it's also particularly complicated because of the different scales that talk to each other, right? You're going to have molecules that are going to influence what's going on in the cell, that make up a tissue, and then you have an organism, and just looking at the molecules is not going to get you all of the way to the organism, and just looking at the organism is not going to be enough to understand which molecules you should be giving to your patients. So what we want to build, what we need to build, and this is what we call multi-model or multi-scale, is a model that can actually look at these different scales simultaneously so that it can understand what the consequences of a perturbation at one scale will be on a different scale. This is what I think in the next 5 years is going to transform the intersection of AI and biology. But we have to get there, and it's not easy. So let's talk a little bit about how we can do that. There are 3 parts to doing foundation models. This is probably generalizable to other sciences as well, but I'm going to stick to my domain. So the first part is data, right? We're talking about foundation models. We need very large amounts of data, right? We're talking millions, billions of data points. If you're thinking about transformer models like Gemini or ChatGPT, the avalanche of data that was necessary, I can't even think about it. It's terrifying. We need the equivalent in the biomedical setting. The good news is that it tends to exist as long as we're OK working with unlabeled data, which is where foundation models shine. And we're going to need them at every scale because we want to build this multi-scale foundation model. We're going to need it for tissue, for molecules, for proteins, for cells. And this is much more, and this is crucial, and I'll be diving into this, but we actually need data that connects the different scales as well, right? You don't want some protein data here and some tissue data here. You want, for the same curl of tissue, molecular expression, right? You want gene expression on that curl that you have an image for. That is much harder to obtain. It started to exist in the past couple of years. That's why we can do foundation models that are multimodal today, in my opinion. But having that data is really like the missing key that was missing for all these years and that is currently transforming the field. The second part is architectures. There was also a great question about architectures. We've seen transformers take the world by storm over the past couple years, right? And that's because they fundamentally have this component called attention that works really well at identifying correlations and like structure within human language, and then has been adapted to work with vision images as well, right? Before transformers, we had CNNs, convolutional neural nets, which themselves were also inspired by biology based on how the human eye works, right? In all of these cases, we have architecture, our algorithms that are built specifically to work on the kind of data that they're trying to work on, right? We're going to need an equivalent for biology. We're going to need to figure out how to make transformers work in that setting, how we need to think about attention, how we can encode the structure in such a way that biological data is messy, right? We want to make it easy for the model to figure out what's going on. And then, on a side note, which turns out to be a major part of the problem as well, whatever fantastic architecture you come up with, which is biologically motivated and super scientific, and you have a lot of like differential equations to explain why that's the best model, it needs to run really fast on GPU clusters. It needs to be able to scale and communicate and have all of these like very low-level hardware considerations at the same time. So we are at this intersection here where we want the hardware to work and the science has to make sense as well. And then last, but definitely not least, and I think this is actually one of the wonderful parts of being in an application to biology, is you actually want good evaluations, right? When we're applying machine learning to biology, to hard science, the burden of proof is really high, as it should be. And so you need evaluations for your models that are not going to just, not that they're meaningless, but they're not going to just be the next NeurIPS paper, my method is state of the art. You need something very specific. You need to go into saying, well, actually, we built this molecule and it works, right? Or here is the setting where our model doesn't work and why. And these are, to me, the major three components to building foundation models for biology today that we all need to figure out so that in the next 5 years, we can build a world that's unrecognizable from today. So let's start with the data. Again, I've said this a lot, a computer scientist. When someone gave me a histology slide and said, oh, you know, we have a couple million, I was like, OK, that's a small number for foundation models, right? Like, it's good, but what are we doing here? And so please bear with me as I talk you through my perspective at looking at H&E slides. This is an H&E slide. It's a stained image of a biopsy. And it's actually not that small. This is the equivalent of some classical machine learning data sets in one image. Because this image is something like 10,000 pixels by 10,000 pixels. You can zoom in. I've zoomed in once. You can zoom in twice. I'm running out of space on the slide, but you get the point. You can look at individual cells here, right? This is a massive amount of data. And we're going to need to build models that can actually work with a setting where one image, you're going to have to try to load more than 5 of them in memory, right? When you're doing batch learning, and I won't go into too many details, but this is a major question. So this is the histology level. This is the tissue. I'm going to be talking about multiple scales. So let's go one down. We start at the tissue. I showed you, you can see some individual cells here. How do we look at data at the cellular level? This is as my team has explained it to me, so I apologize. You have a tissue. You have a biopsy, let's say. The first thing you can do is a bulk measurement, which, as far as I understand, is you take the tissue, you put it, turn it into a soup, kind of shake it about, and then out comes a vector that tells you the number of gene counts per gene. So you have something that's like one vector of 20,000 floats, right? 20,000 counts. It's relatively cheap. It gives you insight into the molecular. It's great. However, I did say there's this soup part. I should stop saying this on recorded talks. But you'd lose a lot of resolution by doing this, right? So there is a different approach, which is going to be looking at single cell level measurements, where you're going to get the same 20,000-ish length of vector, but you're going to get it for every cell that was in the previous slide that we were looking at. All of a sudden, you don't have a vector, you have a matrix. We love data, matrix, more data, we're happy. Obviously, it's more expensive to compute, but you get much finer grained resolution into what's going on. You also get a lot of noise. I mean, you get a lot of noise in biological data in general. I used to generate my own matrices to do math on, so it was a learning experience there. But you do add a lot of noise when you're working in the single cell setting. But we can do even one better. Because when I go from the biopsy to the single cell view, we have this matrix, which in of itself is actually really interesting because it's not the kind of data that most machine learning models work with as an input. But we have lost the spatial resolution. As of a couple of years ago, there are now technologies that actually give you at scale resolution from the spatial image into the cellular. Essentially what it takes is we take our previous image, right? And it's going to tell you, well, see, like this cell here, I'm going to give you the vector of genes in it. And we're going to have this, essentially, we're going to have this matrix that we had for the single cell setting. We're going to pair it to a vector, or a matrix, that's going to give you the XY positions for each of these measurements. This is fundamental, right? Like this is why we can start thinking about doing multimodal within the scope of tissue and single cell. To do this for image and text, you needed the internet where for each image you tend to have a caption that teaches you how to translate from one modality to the other. We need the equivalent in biology, and this technology, this spatial omics, is what is going to allow us to do this if we want to go from the tissue to the cell. I told you that the histology slide is massive. The spatial omics version of it is bigger because it's an H&E slide plus more things. It's going to be like several gigabytes for one slide. It's also expensive to obtain, to be clear. So you have 10,000 by 10,000 pixels, and then you have, I was saying single cell, but often it's also spots. So for the people who actually know what I'm talking, or who have more information than I do about the technology, I do know I was being kind of like hand-wavy with the science. But you're going to have roughly 5,000 spots that are going to give you this genetic readout. When you work in partnership with hospitals, you're not just going to get these two things. I say just, even though they're so meaningful. You're also going to get things like clinical annotations, disease progression, patient medical history. One data point, again, is so incredibly rich. It's so incredibly rich, people have been writing papers on a single data point with reason, right? Like it contains enough information to do that. Now we're in a setting where we can do this at scale, where we can actually ingest this with foundation models. So now let's talk about architectures. I'm not going to go into too much of the details about transformers. You either know how they work, in which case there's no point in me telling you about them, or you know that they are what powers things like ChatGPT and Gemini and Claude and whatnot, and that's what you need to know. Transformers have transformed the way that we think about AI today and how we apply it everywhere. And the question is, OK, well, can we actually make them work on biological data? They were in a lot of ways, inspired to do next token prediction, as was said. It's not always clear that biology is going to look like next token prediction. It can, but not always. So the question is, are they going to work? The answer is yes. I'm here, I'm giving the talk, but you don't need to listen to me. There have been a lot of cool things like the Nobel Prize in Chemistry, right, last year, that is based on like transformer-styled architectures that are like bespoke to the problem that they're looking at. There's a lot of like cool stuff on LLMs and like protein sequences. We were hearing about different technologies right before me. On the tissue, there's no Nobel Prize yet. But there is an extremely rich field of computational pathology where you actually have these foundation models that are based on transformers, vision transformers, as it were, since we're looking at images, that can actually produce very meaningful predictions. They're used in clinical settings. The nice thing on top of that is we have so much data and so many obvious use cases that it's really easy to evaluate them, which I'll be getting to in a second. Then the last part that I personally am keeping track of and I think is just really interesting to be aware of right now is going to be a single cell setting, not only because at Bioptimus we're looking at it, but also because we're starting to see a lot of things like the virtual cell. We're seeing things like models trained on like bulk RNA-seq or on single cell. And to me, that's from my machine learning perspective, but not a computational biologist's perspective, one of the places that's evolving really fast where I think we're going to see a lot of changes in the next couple of years. Because I think we're still trying to figure out exactly how to evaluate these models, how to build them. Again, because the input to this data is not an image, it's not a protein sequence. it's a matrix. And so working with that is actually very interesting, and a lot of technologies are being developed. But the title of my slide, as I sent it, was a very long title that said inside of it, from the tissue to biology. So let me focus on the tissue for a second. I'm going to be talking about one of the models we've developed in-house. It's called Bioptimus 1. I contractually obligated to mention it at some point in this talk. But you can think about it as a generic foundation model that works on histology slides to produce embeddings and then be used for downstream tasks. The way this works, as for most foundation models and transformer-based architectures, is you have an incredible amount of data. You have millions of H&E stained images. They've been digitized, which is crucial for computer science. You're going to pre-train your model, and you're going to end up with what is fundamentally an algorithm where you take either the whole slide that you care about or even just patches, pass them through the model, and get an embedding. An embedding is a long vector. It's a bunch of numbers. But what's important is that once you have that vector, you can do what's called either fine-tuning or few-shot learning, where essentially you're going to say, well, OK, I have this model that's seen a ton of computational pathology slides. I have 100 data points for my clinical trial. I'm going to train a linear model that's going to go from the embeddings of my data points to the actual annotations that I care about. Because you don't have necessarily enough data to do these bespoke models we were talking about at the beginning. You have 100 data points. But you can build a linear model with 100 data points. People do it all the time. And you can build it on top of these embeddings. That's what you call fine-tuning. And then with that, you can do a lot of different downstream tasks. You can do things like stratification, biomarker discovery, and so on and so forth. This is one of the canonical ways that these foundation models are being used today. A slide with numbers. I'm a scientist. I do have to have one of these in here. The point of this slide is not, ooh, look at the top, last right column. It's really good. I mean, it is really good. But the point is, if you look at these are a lot of different foundation models that are on the markets today. What I want to show you is that this works, right? We're looking at here AUCs, our rocks. We're at 0.8, 0.95, right? This is one foundation model, but it's working across indications. Here I picked colorectal, breast cancer, and prostate cancer, but we have benchmarks to the wazoo. So this works, right? We're in a setting where we're getting 0.9, and we're not shocked anymore. It's a really good number. It is usable. You can actually turn this into something actionable if you're a clinician, if you're a researcher, if you're in pharma. But I have 15 minutes left. I'm going to accelerate. I've been telling you about multimodality, and then I said, look at all of these cool things we can do on H&E slides, right? What we want to see is what we can do beyond this. What I showed you is what currently exists. These are models that exist. They're commercialized. If you want to use them, please do on AWS Marketplace. But where are we going to be in 5 years? That's the real question. As I've already told you, in my opinion, it's going to be multimodal. And so the question is, how are we going to actually validate multimodality within this setting? So the simplest thing we can do, because why go complicated if you can go simple, is you're going to take this image that I was just showing you, right? And you're going to just truncate the last part. You're not going to do downstream prediction and all of these things that work based on H&E images. You're going to say, OK, let's be multimodal. Can we actually go to the molecular from looking at the tissue data? So we're going to take this spatial omics, and we're going to say, OK, give me the spatial resolution at the single cell, at the genetic expression. This is work that was not done by us. This was actually a data set and a benchmark that was built at Harvard by the Mahmoud Lab. It's called HEST. And the idea is that they took, and I'm simplifying, oversimplifying, but for each H&E stained image, you're going to chop it into a bunch of really small patches, 112 by 112 micrometers. And there they actually have measurements for the 50 most expressed genes within that very small patch. And so the task is, can you take a model that just looks at the image and is going to tell you, here are the 50 most expressed genes at this resolution? Before I give you the numbers, if you can do this, spatial omics is incredibly expensive. I'm keenly aware of how expensive it is. If you can obtain them by passing them through a model, it's much faster. there's a lot of work in scalability and so on and so forth. So that would actually transform medical research and so on, diagnostics, right? Like getting a biopsy, it's not pleasant, but we know how to do that, right? Spatial omics is still very, very new. If you can just pipe an H&E slide into a model and say, well, here's actually what the spatial omics look like, we're in a different world already, right? We're 2 years into the future. So can we do this? Once again, there are two ways of looking at the numbers that I'm showing you. One of them is looking at the most left and saying, like, wow, H-Optimus 1, doing really well. The other thing you can say, though, is as a scientist, you look at it and you're like, OK, these are Pearson correlations. A Pearson correlation of 0 is random. A Pearson correlation of 1 is perfect prediction. We're at 0.4, right? We're not hitting 0.5. We're not, nobody else is. We're in a scale where we're definitely doing something useful. Don't get me wrong. This is the setting where we can be involved within experimental design, for example. Maybe you don't want to do spatial omics, but you might want an IHC panel. This can help you kind of figure out the space of possibilities. But we're not at 0.1. And we're scientists. We'd like to see 0.1. 1. We want to see 1. That's the number we're looking for. That's when we know that we've actually built a model that captures the different interactions. So let's zoom into this for a second. This is exactly the same plot as before. And what I'm going to do is I'm going to look at what happens if we just stratify these results across cancer indications. So this, I'm representing the results for H-Optimus 1, but again, like this generalizes across the different models. We just have the numbers specifically here. So here are across different indications. All of a sudden, the picture is much more complicated, right? For example, for skin cancer, we are hitting something like close to 0.7. 0.7, we've gone, now we're talking, right? 0.7, you're much more interested all of a sudden in like saying, OK, I'm going to use this. You're not 1. You can't replace spatial omics. But you can definitely think really closely about how badly you need them. On the other side of the curve, you have rectal cancer. And there, you have 0.2. Smaller. It's not as good, right? And so here's the thing. We want to understand why we're not hitting 1. We have the data. Foundation models are supposed to be really good. What's missing? These results, we see them across all of the computational pathology models that exist, right? Like this has been benchmarked independently by the Mahmoud Lab and others, which leads me to believe, and this is my thesis and where the next 5 years are going to be, that the problem is we trying to fine-tune a model into being multimodal. Right? We took a model that was pure histopathology, and then we said, OK, now do spatial omics. What we need to do, I would argue, is from the ground up say, here's a model. It understands all of these scales at once. It was trained on all of them simultaneously. This is something, again, it's very new. It's cutting edge because we didn't have spatial omics until very recently. And when I'm talking about spatial omics, I mean at scale, right? At the scale that you can use for foundation models. So how are we going to get there? This is the somewhat speculative part. It's also the part where I can't give you details, unfortunately, because this is proprietary technology. But I was talking about 3 things, right? Data, architectures, validation. Guess what the next part is? Data. Great. So we need multimodal data. There is multimodal data available online that is open source. There's a lot of like great, I think someone mentioned TCGA earlier. That's like one of them. You have like, and then there's H10 that's also available online. So there is multimodal data online, but you need a much bigger scale. So at Bioptimus, we have, of course, a ton of H&E images. We have built these foundation models for H&E. We also have data acquisition programs focusing on generating and supporting essentially the generation of other data that supports within this. There's H10, which is, again, lovely resource. And then we also benefit from the Mosaic initiative from one of our partners, Oken, which is a partnership with hospitals across the world that generates multi-omics, spatial omics, H&E, WES, clinical annotations, and everything else that's also on the slide that I'm not going to just read out to you. This means that we're at a setting, right, where it's becoming possible to have something that is really natively multimodal. Next part, architectures, right? So this is this part where I'm going to be very nebulous and not go into the details. But when you're looking at things like spatial omics, when you're connecting the image of the tissue with what's happening at the molecular level, right? you actually you're giving so much information to the model. You really need to help it along a little bit, right? I talked about CNNs and transformers. We need to build the equivalent for our setting. This means attention mechanisms that can think about what's going on at the spatial resolution, what's going on, like, these are built on curls of tissue. Curls of tissue are, like they have a z-axis, right? Like, how do you take that into account into your modeling, for example, right? How do you connect that with spatial expression? How do you connect that with the noise and the data that comes from spatial omics? You're going to need to model all of that very carefully. There's this excellent question earlier, right? It was like, how do you actually build models that are going to work in this setting? To me, it is the question, because you can't take Gemini and fine-tune it on this data. It doesn't work, right? We need to be bespoke. And then there was a slide that said, and then the benchmarks. But what I was going to say about the benchmarks was essentially, well, they're still very nascent, right? So we have HEST. We're working in collaboration with MIT right now. There's this ARPA-H program where they're working on like foundation models that are going to be multimodal and able to benchmark on clinical data on patients that are being measured right now. But it's still very nascent. And us and everyone else in the industry, but I'm the one talking to you right now, are looking also for collaborations in places where you can actually build meaningful benchmarks of multimodality, as opposed to, we tried, like, here's data that's already available. We want something that's going to be directly useful for research, both academic and pharmaceutical. So with 7 minutes left for questions, I think that what we're going to see in the next 5 years is going to be multimodal foundation models. But that means that we need access to both incredible, vast amounts of extremely rich data, and we especially need to learn how to craft the models that will inhale that data. And that's what I hope to see.
So that was a very computer science-y talk. I like it, being a computer scientist myself. And it's hard to connect computer science to biology and present it in this kind of interpretable way. So kudos for that. All right, so please go ahead. We'll start with you.
Thanks for the talk. Is modeling the temporal aspect part of that multimodality, especially embryonic development and the subsequent development of a human organism?
Short answer, yes. Long answer, it's complicated. Of course, the temporal aspect, right, is why it's one of the reasons why we actually care about doing this, right? We want to look, for example, the study we're doing with MIT is looking at progression-free survival. The temporal aspect is what we're all trying to solve. The thing is, getting temporal data is possible, but it's hard. It tends to exist very much in the annotation space, right? You're going to have, the patient was administered this drug, we saw this response, they tried other this, like they have this, and medical history and like family history is also temporal data. When it comes to things like H&E slides, I was actually told by an oncologist very grumpily, like, please stop asking for patients to get biopsies every 5 minutes. It's not an enjoyable process, right? So there it's more complicated, it does exist, but it's going to depend on the indication, and it's going to be in much smaller resolution. So there's actually a lot of really interesting work and modeling going into how do you, same as I was just talking about, take this very comparatively small amount of temporal data and enrich the model with it.
Go ahead.
Hi, thank you so much for your great talk. I'm Nazanin Ahmadi from Brown University. Since you are a computer scientist, I have a very computer scientist-like question for you. Have you ever tried using Mamba state space model or visual form of it, like visual state space model, vMamba, to replace your transformers? Because one challenge using transformers is like their high computational cost, but Mamba doesn't have that problem. And in my experience, Mamba outperformed the transformers.
I want to follow up on that because that's actually really interesting. I think I can answer this. We haven't. We also have access to enough GPUs that it's not our first axis of optimization is going for something that's like lower consumption power. I'm actually curious, like what you've compared Mamba to. Is it things like DynoV2? Is it different? But I can ask follow-up questions afterwards, but
Yeah.
I'm actually really curious about how you looked at those two.
You know, I use it not for like images, but for like, I don't know, the prediction of treatment schedule for tumor weight, something like that in the setting of neural operators.
Okay.
But I know, yeah, they work better than transformers in terms of the number of parameters that you are trying to train, in terms of the extrapolation, in terms of prediction, accuracy, and so on.
Okay, no, that's very interesting.
Yeah.
In images and like in the image modality, I do think like right now vision transformers are kind of state of the art. But again, I will find you after this.
Thank you so much.
Do we have any questions online, Wingo? Yeah.
So we'll take the next question here.
Oh, you want to?
Yeah, sorry, I wasn't obvious.
Sorry.
Hi, I really like your talk. My name is Yo-Yo. I'm a postdoc at Northeastern University. I'm also co-founder of Dark Matter Therapeutics. I'm a computer science too, so I really relate a lot of things you said. I really like how you formulate this into this multi-scale, multi-level, multi-modality integration, because I think this is what we really need to solve the biology problems. So my question is also like during practice of like, I also want to do things like this very similarly, but the things that are so many like incomplete modalities, incomplete data, you know, like one very obvious example is what I've been doing. So let's see, we have a lot of like spatial transcriptomics. However, like the resolution of spatial proteomics, like I think we can only cover like thousands of them. So like how can you, or do you have any thoughts of like addressing that kind of like more in general, it's like this missing, you know, missing data and missing, you know, modalities? How do you really, or any thoughts?
Thank you for the question. I would have planted it if I could have. It's a great question for Bioptimus specifically, because one of the things we're looking at is actually generating modalities from existing modalities, right? So you're saying like if we have enough data to connect some examples of like spatial transcriptomics to spatial proteomics, then the question is like, can we then generate spatial proteomics, right? So that is actually one of the major things we're looking at as a use case, as well as a validation, right? I was talking about how important it is to actually validate if your models learn the connections. This generative approach is one of the easiest, with in heavy quotes, actually ways of validating this, because we can then confront it on a small set of measurements to the real thing. The other thing, I'm bouncing off of your question, but actually the different resolutions is also like a really fascinating question from a modeling perspective, because even like H&E slides, right? Even without going to the spatial transcriptomics, spatial proteomics, which indeed are like very different resolutions, and it's a whole thing. The slides also exist at different resolutions when you're accumulating them across centers, et cetera. And so you actually need to consider how to train models that can adapt to the different resolutions. This is a related question, but it's an interesting thing, I think.
Yeah, I think we can discuss later, but this is a very important question to address. Thank you.
So let's thank our speaker one more time. In the interest of time, we'll move on.